Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for UW Extension Cooperative Extension. And on the behalf of those folks and our other co-organizers, Wisconsin Public Television, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you David Baum. He's a professor of botany. He was born in London, England, and he went to high school on Kingston upon Thames, and then he went to Oxford University where he majored in botany. Then he came to Washington University in St. Louis to get his PhD in evolutionary and population biology. He did a postdoc here at UW-Madison, and then he went to a faculty position at Harvard where he got tenure, and then he decided to come back here for the ice fishing. And and came back to UW-Madison in 2001. Tonight he's going to talk with us about the origin of life as a chemical ecological problem, new ideas and new experiments. Please join me in welcoming David Baum to Wednesday Night Live.
Well, thank you, Tom. Thank you for inviting me to this. So, the origin of life is usually framed as a problem of chemistry and physics, maybe a little bit of planetary science and geology. But what I'm going to try and argue today is that to really understand the problem, you have to also integrate ecology and evolutionary theory into your thinking. And that when you do that, I believe that you'll start to see that there's a whole class of experiments that ought to be done that haven't historically been done, which have the potential to really help us understand how life originated on this planet and where else in the universe we might find life. So what I'm going to do is I'll kind of introduce you to my thinking, and at the end I'll tell you about these experiments, which we're just starting, but I'm very excited about them, which is why I'm so excited to be here and tell you about it. So the way that I want to get started is to start from sort of a more conventional view. So the conventional way of thinking. Do you want me to start again? No. Okay. So the conventional way of thinking about life and how it evolves is from the perspective of cells. So all life we know of is cellular. And cells are very clever little things that can take food from the environment and energy from the environment, grow and divide, and therefore reproduce. And we understand very well that if any variant cell is better at surviving and growing and dividing, that variant will get enriched in the environment. It will be preferentially passed on. This is natural selection. And that's a very simple concept, and it explains how it is that once you have cells, complexification can occur. Because natural selection doesn't bias against complexity, and in some situations it can favor complexity. So we fully understand that once you have evolving cells, even of a very, very simple kind, they will complexify to give rise to the amazing organisms we see around us today. The problem with this view is, how do you get started? Because if you take the view that you need a cell to evolve adaptively, and adaptive evolution is the only way you can generate complexity, where does the first cell come from? You're forced to the position that the first cell arose already able to function, already able to undergo adaptive evolution. And I would like to question whether that is really a plausible proposition. So think about what even the simplest cell needs to do. A cell is a membrane, and this membrane has to contain a certain set of key metabolites, key chemicals. And those chemicals interact with one another to take food from the environment that has to be able to pass through this membrane, and convert that food into waste, and in the process generate more of those metabolites so that the cell will grow. And all that has to happen without any prior. In order for evolution to start, this has to happen spontaneously for the first of these functional cells. And then, to make it matters worse, this cell, this first dividing, growing cell, after it divides, has to give rise to daughter cells that are at least as good as the parent cell. Because if they're both worse than the parent cell at growing and dividing, then over time they will degrade into non-functionality. So I view this, and I'm not alone in this, as a very challenging proposition. The idea that without any adaptive process, a cell that is functional, that is able to evolve, could arise spontaneously, could happen. I think that's very improbable. So we need alternative models. So the alternative model that I think you may have heard of is the idea that the first replicating entity was not a cell, but it was a single molecule, a single very talented RNA molecule. As many of you will know, RNA molecules are kind of interesting molecules because they can template information, but they also can function as catalysts. And so the idea is that if you had a single RNA molecule that could replicate itself, then it could give rise to a population of descendants in which natural selection happens. That's all very well, but it also poses a lot of problems. Okay? So the first problem is, where would this very long RNA come from? In natural environments, RNA is not stable. We have to work hard to maintain RNA stably in the lab. In a natural environment, it would take a lot of energy to generate these RNA molecules. But supposing that a long RNA molecule was formed and happened to be able to replicate itself without any prior history of adaptive evolution, how would it run that? In order to be able to replicate itself, it needs an input of energy. For example, it would need activated building blocks, nucleotide triphosphates. Where would they come from? And how would it be able to harness those indefinitely? Okay, supposing that problem were overcome, it would then have to be able to copy itself actually accurately enough that its descendants, its copies, have a high probability of being as good as it at replicating. And a lot of theoretical work going back into the 70s has shown that this is actually very hard to imagine, given what we know about how accurately an RNA molecule can function as a catalyst. And lastly, even if we accept that all these things could happen, how would this RNA molecule build a cell around it? That is not really at all clear. So the bottom line is that if we rule out the idea of the first thing that could evolve being a cell, I would also add we can also rule out the first thing that could evolve adaptively as being a single RNA molecule. So we need yet another approach. And to see that other approach, I think we need to sort of take a step back and think about what life is. So, how do we define life? Well, there are many definitions out there, but the most popular definition right now is the so-called working definition. This was developed by a committee at NASA, and it defines life as a self-sustained chemical system capable of undergoing Darwinian evolution. So I'm going to sort of break this down. I'm going to first talk about the self-sustaining part, and then I'll talk about the evolution part. So what does it mean to be a self-sustaining system? So what this asks us to do is, instead of looking at a cell as a single entity that can grow and divide, think of all the chemicals that make up that cell. The set of chemicals that make up a cell are of many different kinds, but they have the property that they cooperate. They work together to use stuff in the environment, food and energy, to make more of the same set of chemicals in roughly the same proportions. So that as a cell grows, all the chemical species in there all double ultimately until the cell can divide. So what's happening then is we have an ecosystem in a cell, a bunch of different species that cooperate and interact with one another to generate more of the same, and therefore to grow. So we can think of a cell, any living system, as a chemical ecosystem. So as I said, modern cells can be thought of as these ecosystems, but they're not like the ecosystems we see in nature, because they have cells have a membrane around them. They have a natural edge. If you go out and you find an ecological community, there's not an edge around it unless we put it there as humans. So, but despite that, you can imagine a functioning, life-like chemical system that didn't have a membrane. And to help you visualize this, think of primary succession happening on something like a sand dune. So when a sand dune is colonized by the first few plants, so we've got a few little straggly grasses here that have managed to take root, well, they form a nucleus around which other members of that species and other species can start to collect because the sand is now being stabilized. And this community, which can nucleate at one point, can grow outward. And the notion here is that this community can function and grow even without having a boundary around it. Could that be true for primitive chemical systems? And the answer is that in theory, it could. So a lot of theoretical work over the last, especially decade, has shown that if you have a mineral surface or a solid surface and a subset of chemicals become associated with that surface, sort of like what happens when these plants first colonize a sand dune, they can nucleate, and if they cooperate with one another, they can make more of the same chemicals in their vicinity. So they can spread and grow over the surface. Now, the little acronym I like to use for these systems is surface-associated lifelike interacting molecular ensembles. So that makes it slime, okay? Slime. So I'm going to use the word slime because it's catchy. So slimes are chemical ecosystems that are unbounded but can nonetheless grow and function like a cell in that they can use material in the environment to make more of the same chemicals and expand. So to help you visualize this, here's sort of a cartoon example. Imagine we have this mineral surface in kind of gold, and there are these 4 different kinds of chemicals, big A, big B, big C, and big D, all of which have the property of sticking to this surface. But they're not present in this solution, in the ocean. But very rarely, they can be formed by conversion from a precursor. The precursors being little a, little b, little c, and little d, which don't stick to the surface. Now these chemicals, I call them keystone species, which is an ecological term, form a catalytic loop. So for example, big A helps little b make big B, and big B helps little d make big D, and big D, I've lost that, big D makes little c make big C, which helps little a make big A. So you have a little closed loop, which means that if this is sitting in an environment in which little a, little b, little c, and little d are flowing in the ocean, then this system, once it nucleates on the surface, will grow and spread over the surface. It's sort of shown here. And this growth and expansion will continue in principle until either one loses the monomers, the building blocks, the A, B, C, and D, or the surface is full up or destroyed. So this is now how I think of the very beginnings of life, is of these slimes forming spontaneously on mineral surfaces. And where do I imagine it might happen? Well, there's many possibilities, but the most sort of obvious place to think about would be something like a hydrothermal vent. So hydrothermal vent is a place in the oceans where water has percolated down into the crust, into the rocks, and it gets forced back up to mix with the ocean after having been changed chemically quite a lot. So what's interesting is that at hydrothermal vents, there are a lot of chemical reactions that convert carbon in the form of maybe carbon dioxide or methane into some of the small building blocks of life, simple organic molecules. There's also a lot of chemical potential energy here because the vent fluids are in a very different energetic state, a very different redox state to the ocean they mix with. And so there's energy here that in fact many living organisms, many living microbes use to grow and live. So, but they could also, this energy and these little food substances could in principle promote and allow slimes to form and then grow on these minerals around hydrothermal vents. So that's how to think of a slime as a self-sustaining and growing system. But what about evolution? in order to be living by my criterion, by that definition, it also has to be able to evolve adaptively. Can they? Well, I'm going to suggest, and there's a lot of emerging theory to support this claim, that they can. They would, in fact, evolve. So the first thing to be aware of is that as a slime is spreading over the surface, it's carrying with it the ability to continue growing. So there is a kind of heritability. So as it grows, the ability to grow is carried forth in the slime. So that is a form of heritability. And if for any reason one part of the slime was different than the other parts, and that part could grow more rapidly, we would expect that part to take over and to maybe cover more of the surface in the long run. Okay, but how might variance arise? In order to have adaptive evolution, you need heritability, which is to say a sort of maintenance of some kind of character through time, but you also need a way to generate new variants. So what does that look like? We don't have a genetic system here. Well, again, the modeling suggests that we should understand this in terms of network evolution. So autocatalytic chemical systems, of which slimes are an example, are composed of little autocatalytic loops. And in principle, there can be multiple of these loops that can exist. Each of them is called a core, an autocatalytic core. So in this little example from this paper, there is this one sort of greenish core, which is composed of 5 keystone species that form a loop. So if those species associated with the surface, it would be a slime that could grow over the surface, provided it got those food inputs. But if, sometime, by chance, a little bit of A and B were produced in the same area, now that also forms a second autocatalytic core that can be added to the first one, and the two can actually coexist depending on the details of the model. And so these slimes can actually evolve not through genetic mutations, but through the addition of these autocatalytic cores. And as a result, they are, in principle, evolvable. So the take-home here is that, at least when one approaches this from a theoretical point of view, these slimes, which sound like they ought to be able to rise kind of easily, would begin adaptively evolving right away. And specifically, they would, over time, you would expect them to get better at colonizing new surfaces, at resisting disruption, and competing with other slimes. So we would expect some sort of early complexification well before you have cells. Now, if you remember when I talked about RNA, one of the weaknesses was that there wasn't a simple way to go from a self-replicating RNA to a cell. Because we know, after all, that cells did eventually evolve. So we have to, this approach, this kind of ecological way of thinking about chemistry only helps us if it provides a path to the cell. So can my slimy model explain the origin of the first evolvable cell? And the answer is that it does so very naturally. So to kind of talk you through this, imagine the following scenario. We have an ocean with patches of a certain kind of mineral that is suitable for slimes, and 2 different slimes evolve independently, one in green and one in yellow. Now, those every so often minerals are going to become unavailable. They'll get eroded away. They'll get covered by something, and also every so often a new virgin mineral patch will appear due to a disruption. Now, if a certain slime happened to produce a little piece of itself and release it every so often into the water column, this is a little dispersal unit or a propagule that would get up into the water column, it would allow that particular kind of slime to spread and colonize all these mineral surfaces. And so it would gain an advantage. So over time, you would expect to see more of the green and fewer of the yellow slime, because it can keep colonizing as new environment becomes available. Furthermore, if there was a mutation that allowed that propagule, which is just an inert blob of protoplasm to actually be able to do some metabolism in the water column to take in food and grow a little bit, let's say just a little bit of growth, maybe you can see that, that would give it potentially a competitive advantage because it would be able to colonize more effectively on a new surface. The same principle, selection for enhanced colonization ability, would then favor a variant again that was able to divide. So now in the water column, it can do metabolism, can grow, it can divide, and it can colonize multiple mineral patches. And once you have a propagule or a blob that can divide on its own, then we don't need to go back to the surface, and we have basically an evolvable functioning cell. So in summary, from this perspective, what's nice about the slime model is that it actually provides a very natural selective stepping stone to a cell with division being the ability to divide being a relatively late addition in that sequence. So this model represents a change in concept. And I think the way that I would most starkly put this would be that it really suggests that when we think about the origin of life, the traditional view puts it in rather stark terms. That we go from non-living systems to the spontaneous appearance of something that has to be quite complicated but can now evolve adaptively, a cell, and then we have life. That life is a gulf that at some point was crossed. The perspective I'm trying to get across is a much more gradual transition from very simple chemical networks, chemical ecosystems that can colonize and nucleate on mineral surfaces and grow, but then can gradually complexify, transitioning from sort of being unbounded slimes at some point to being cells, and eventually to adding this familiar machinery of living cells, which is to say DNA and RNA and a genetic system. So this transition is a much more gradual one in my way of thinking. Furthermore, it raises the possibility that at least the early stages, these initial transitions, might occur easily and quickly, rather than requiring a long wait for a miraculous transition. So taking this view, we can now ask, could slimes actually arise easily? Because I think you can see that if slimes could arise easily, suddenly whole new research programs become possible. So I'm going to suggest that while we don't know that slimes could arise easily, there's nothing we know that says they couldn't. Not a great argument, but it works, okay? So the first thing to bear in mind is there is no evidence of a period when Earth lacked life. So the oldest rocks that could contain direct, tangible evidence of life do contain evidence of life. Now, that doesn't mean that there wasn't a long period after the planet formed when there might not have been life. Because for the first few 100 million years that the planet was potentially habitable, we have no rocks to give us a record. It could be that life of some kind originated, 3 days after the planet had water. It could have taken a couple 100 million years. We don't know. But it's at least possible that it happened quickly. A second consideration is you might argue, well, if life arose easily, wouldn't we be aware of many independent origins of life? Whereas all the data clearly show that every life form we know about today traces back to the same origin. If life could originate easily, wouldn't there be many origins? Actually, Darwin himself pondered this question, and he pointed out, genius that he was, that if once you have very able, complicated cellular life around, it's going to make it very hard for any other life to actually become complicated enough for us to see it. Because life will eat it. Life will eat its food. Complex life will preempt simple life becoming complicated enough to be seen. And then the last thing to be aware of is that actually we don't know that there aren't slimes all over the place in the environment, because we don't have a good way to detect them. So the upshot of this perspective is we don't know that slimes can arise easily. We don't know that these evolving systems can arise easily, but we also don't know they can't. And the only way to explore this as a scientist is to get in the lab and try and do some experiments. So that is what we want to do. So this is where I transition to telling you what we're hoping and trying to do in my lab and in the lab of a number of collaborators around the country. So the hypothesis that we want to test is that slimes, which is to say chemical systems that can colonize a mineral surface, can grow and potentially evolve, can arise quite easily. So how could we detect them? So as an evolutionary biologist, it seemed obvious to me that we should go right to the heart of the matter. If our definition of life is something that can evolve, we should be looking for things that can evolve. So what we should do in our experiments is impose artificial selection for some characteristic on a chemical system that might contain a slime, and then see if there is a response to that selection. If there is a response to that selection, that will point to the existence of something that could propagate itself and evolve. So those are the experiments we're trying to do. So let me talk you through, this just really repeats that we want to impose selection on mixtures of chemicals and then use the response as evidence that there is, in fact, a lifelike chemical system present in that mixture. So how are we going to do this? So the first thing we need is some kind of chemical soup. We need a mixture of the kinds of small molecules that could potentially form the keystone species of an autocatalytic system. Now, what we've been doing to date is creating ourselves, buying from Fisher or another company, individual chemicals and mixing them together to make our own sort of prebiotic soup. But what we can do and hope to do, thanks to a collaborator in the future, is actually take the soups created in Miller-Urey type experiments. So Stanley Miller and Harold Urey did these experiments in the 50s where they sparked primordial atmospheres and they showed that you could get liquid soups that contained many of the building blocks of life. And so in the future we'll actually be using real Miller-Urey soups, but right now we're making our own from scratch. The next thing you need is some minerals, and there's lots of minerals to use, and we're using the experiments we planned this summer. We'll use 4 different minerals, but one of them is pyrrhotite, which is an iron sulfur mineral that was abundant on the early Earth, and some theories from the 80s suggest it's a very good candidate surface for the formation of one of these autocatalytic systems. The next thing we want to do is add some kind of energy, because in order for creative work to happen, in order for a system to be able to maintain itself, it has to be an input of energy. And what we are using as energy, either redox disequilibria. So for example, we might add ammonium and nitrate. So these are the ingredients of a fertilizer Baum. So there's a lot of chemical energy, but it's chemical energy that is not easy to activate. So that would be one thing we could add. Another thing we could add would be high energy phosphate bonds. That's the kind of energy that our cells use. So we actually, in some experiments, add ATP, which is a source of phosphate of energy. And then once we have all these pieces in place, we can then select among a set of cultures or tubes, vessels, for those that use the most energy. So let me kind of talk you through that because it may not be obvious how this works. So we can take a plate with 96 little wells in it, and we can put minerals in every well, and soup, and our energy sources. And we can incubate it for a period of time. Then we can measure how much energy was used up, or conversely, how much energy is left in the well. And we can identify by a colorimetric reaction which wells had used the most energy. We can then take the grains out of those wells, mix them together, and feed them into a new plate, along with virgin grains of the same kind, and new soup and new solutions. And then we can incubate again, and again select. Every generation we select for whichever wells, whichever cultures have the strongest response, the strongest signal. And the idea is that if we do this for many generations, we will see that the mean value of the trait, the mean amount of energy used, will gradually change. This would indicate a response to selection, which is hard to imagine how you can get a response to selection unless there's some heritable system that is being preferentially passed on during the selection process. So these experiments are just beginning, so I won't give you any results. We've just got all the equipment in, and we have, I have 4 students working over the summer who are going to be cranking through a bunch of these experiments. There's a second approach that we've already tried, which is a little simpler, but maybe a little less direct. And this is a natural selection variant. So in a natural selection variant, we exploit the fact that if you serially transfer from one vial to another vial, a subset of the mineral grains in the first vial, if you keep doing that many, many times, you are inadvertently selecting for any slimes that might be present that are good at getting from one grain onto another grain. You are selecting for the ability of systems to survive and spread from grain to grain. Because every generation you only take, let's say, 10% of the grains to move them to the next generation. So if you have a system on those 10% that can spread onto the other 90%, then those will get enriched over time. And because these are chemically active systems, you would expect them to change the composition of the soup. So what we can do is measure various features of the soup. And so the thinking is that, let's say we did have some of these slimes, let's say a red slime and a yellow slime, and the red slime maybe isn't quite as good as the yellow slime at getting from grain to grain. Every time we take a subset of the grains, and over time we'll eventually dilute out the red ones, and we have only yellow ones left. And since the yellow ones are going to have different chemistry than the red ones, we would correspondingly expect to see just changes in the chemistry of the solution after the incubation. So here are a number of just pictures. These are quite simple experiments, and I've been blessed with a great team of students who've been working on this, mainly undergraduates. Actually, I see one of them in the room. So this is, making solutions. These are the vials we incubate them in. This is actually grinding up the minerals. This is working in an anaerobic chamber to avoid the minerals becoming oxidized in air. This is an incubation happening. So these are relatively simple experiments, and we've done this one experiment we've done for 100 generations. And what we hoped to see would be a trend over time. Something like this. This is actually the result of these experiments are very similar to experiments that are sometimes done on microbial communities, which is what this is from. But this is what we found, which is we had 10 replicate lineages. None of them showed a trend over time. So this tells us that we haven't got the right conditions, if indeed we can find the right conditions. There's another thing we have been looking at, which is maybe a more sensitive indicator that we have something interesting, which is to see if our systems show heritability. So what this graph shows is there are pairs, generation 48, 49 for experimental replicate 1, 48 and 49 for experimental replicate 2. Now, if these systems are heritable, then generation 49 will resemble the character of its ancestor in generation 48, its ancestor being the vial from which we took the grains. And so you might expect to see a positive correlation between the values in the two generations, and that would indicate that there is something in the vials that is carried from generation to generation and affects the chemistry. However, in this case, while there is a very slight net positive slope, this is not significant. So as of right now, these experiments haven't yielded any evidence of a slime, but we have a lot more conditions we want to try. We have a lot of ideas for interesting soups, interesting energy sources, and interesting traits to measure. So, so these experiments are just getting underway, and we have some new approaches that we plan to take in the future. I just highlighting here a student who designed a very beautiful little device that we may, hoping we're going to be rolling out to start working with in the future, which does kind of a natural selection type experiment. It's called a packed bed flow reactor. So the question we have to ask is, well, if it works, it would be really exciting. Okay. Now, okay, I'm going to be a bit over. This is a bit over the top. Okay, so yeah, it is just slime. And so I think we should pause a second and say, this would not be a case of creating life, because if this works, this indicates that these kinds of systems are formed all the time. It wouldn't be creating life, but it would be detecting these very simple earliest forms of life, which I do think would be really exciting. Not in that kind of crazy sense, I hope, and I hope I won't get carted away. So the first thing about this is that we get the opportunity to actually test the theory, because the theory is great, but wouldn't it be great to actually have slimes, to have these self-propagating chemical systems and see if they can evolve and see how they evolve. We can also ask interesting questions about the extent to which the actual chemistry we find in these systems resembles the chemistry of life. Is life the way it is just because it had to be this way, that every time life evolved on a planet like ours it would have our kinds of chemistry? Or could it be different every time? Could there be chance that determines exactly how these chemical systems work? Along the same lines, we could ask how repeatable are these phenomena? And interestingly, we could ask what conditions are capable of yielding slimes or lifelike systems and which conditions aren't. And that could give us really nice clues as to where else on the planet, where else in the solar system and the universe we might expect to find these systems being initiated. So this project is now funded by NASA, and we call it the approach we call chemical ecosystem selection. So it's a chemical ecosystem selection paradigm for the origin of life. NASA likes catchy acronyms. This is a cesspool project, and obviously we're looking for slime in our cesspool. And so this is a multi-institution effort that aims to develop these chemical ecosystem selection methods, obviously look and hope that we can find evidence of these lifelike systems. But even if we can't, our hope is that we can create protocols and devices that we can distribute to many, many labs and begin kind of a more systematic search. Because the number of chemical soups, the number of minerals, the number of environments that you could try is infinite. And in the same way that it would be nice that, that SETI, uses many citizen scientists to search in data sets, we like the idea of being able to get many labs on board searching for a set of conditions that generate these systems, even if we can't find them right away. So that's our image. This is the consortium. So we're the lead institution, but we have 7 other partner institutions, all of which are doing variants on these experiments or developing models and theory to back them up. So I am excited to see where this leads, and I hope you are too. But of course, we wouldn't have got here without a lot of people doing a lot of useful things. So in addition to my collaborators in the Cesspool Consortium, I have a lot of colleagues here who've been terribly important and useful for ideas and techniques and support in the lab. I have a wonderful lab. Mainly, I want to particularly pick out Lena Vincent, who's my graduate student, who's been doing a lot of the work, kind of getting us geared up for these experiments, and a big team of undergrads. So these are all undergrads or one special student in there. And then collaborators and our funders, NASA, NSF, and thanks to my department and the Wisconsin Institute for Discovery. And lastly, thanks to you for your attention.
You're doing these transfers and assuming that the slide that you have will go on. Do you do any kind of pre-screening on the chemistries? I mean, for example, I can imagine that perhaps by making a sort of smooth slice of the mineral, you could add some of these chemicals together and see if they're actually creating a surface-associated layer.
We don't do exactly that. Yes?
Could you repeat the question?
Sorry, good point. Thank you. So the question was, could we do any pre-screening where we would determine which chemicals might actually associate with the surface? And you said maybe we could have a single big surface and just see what happens. We are doing something similar, which is we are taking our soups, and in fact I got data just this afternoon, by taking a whole soup, adding minerals, incubating it, and then just seeing what changes. Using mass spectrometry to really look at all the chemical species. We don't know what, so through that, we have determined that there are, like for example, many of the amino acids do seem to stick to the surfaces. We add the minerals and they very quickly seem to disappear, but we think they're on the surfaces. So yeah, that's the kind of, there is a lot of background legwork we can do, but at the end of the day, we haven't figured out a way to tell us it will work except by seeing it work. Yeah?
I was wondering, have you done anything about miniaturizing your experiments so that you can do a lot and figure out exactly what you want to measure?
So the question was, have we thought about miniaturizing our experiments so that we can consider a lot of conditions? We have. So that reactor that I mentioned at the end is one step in that direction. This is a 3D printed device that is quite small, and in principle we could run in parallel a number of experiments, but we're still talking in the tens. We had a proposal that was unsuccessful to build a microfluidic device that would be able to go and do potentially thousands or hundreds of these at once. That wasn't funded, and there are problems because once you get to the very small volumes, the analysis becomes very difficult. So we haven't yet found that balance, but in the long run, absolutely, I want to be able to make it easy, cheap, and scalable. Yeah?
Where does water fit into this?
So the question was, where does water fit into this? We are doing all of our experiments in an aquatic setting. We're using aqueous solutions. And in principle, there's nothing in this setup that would require you to do that. If you chose to do these experiments in oil using different chemicals and different minerals, you could. The reason we are sticking with minerals like are found on Earth, water like we find on Earth, chemical building blocks like we find on Earth, is because we know that it happened once on Earth. And so I take the view that if this does work, there's nothing, I am not convinced that there's something magical about water. I mean, I know that that's, the wisdom out there is that you have to have water to have life. I'm not convinced that you need to have water to have a life-like system. It just might be very different chemically from us. Yes?
How did you come up with the idea of 100 generations? It strikes me that maybe you need 1 million.
Yes. So I don't know if I need to repeat that, but why did we pick 100 generations? So that is, that's arbitrary. I'll be blunt. Now, in the artificial selection, there's reason to think that you shouldn't have to wait that long in artificial selection, because if you can't detect it in 100 generations because of the compounding way that artificial selection works, you'll never see it. But for a natural selection experiment, you're quite right, 100 generations might not be enough. And just to be honest here, there is a big problem that we haven't yet figured out how to answer, which is how long should a generation be, and how much should we transfer between generations? So let's say we decide, as we have done, that we have 2-day generations, and we transfer 10% of the solution each time. That would require that the slime, on average, be able to multiply tenfold in 2 days. And that may be unrealistic. We may be diluting our slimes out of existence because we picked the wrong generation time and the wrong dilution rate. So there are these problems that we don't have a magic bullet way to solve, but we're going to keep trying.
Does temperature play into this? I mean, have you looked at different temperatures? I mean, you're using a chemical energy input.
Yeah. So the question was, what about temperature? So the experiments we've done have been at room temperature for convenience. We're now moving to some higher temperature experiments for 2 reasons. First of all, as I think you're intuiting, if we're worried about the rate, then temperature generally increases rates. The second reason is that when it's hotter, we potentially make it easier to keep things sterile, because life is pretty good at getting into things, and it would be a false positive. So we are playing with higher temperatures, but in principle, there's nothing magical about any one temperature. Yes, at the back.
Are other institutions pursuing the same basis of pursuit as what you're doing?
So I showed a moment ago the list of what happened was very fortuitously for me last year, the NSF and NASA teamed up on an Origin of Life initiative, and they did something called an Ideas Lab. And an Ideas Lab is where they select 30 scientists from around the country, and they locked us away in a kind of crappy hotel in Baltimore, near Baltimore, and they said work together and come up with new projects. And I already had this project in mind, but I managed to convince these other guys that this kind of approach was the way to go. And a number of them have some really excellent expertise that's relevant to this. Mike Travisano, for example, he is sort of one of the real experts at artificial selection in living systems. And Niall Lehman is also. So basically, yeah, it's now a multi-institutional collaboration, which is great. Yeah, back there?
And are these institutions using the exact same process that you guys have developed here at UW, or is there some freedom to try different things?
Yeah, people are trying some different things. So if you're interested, these two are theoreticians. Niles is actually doing an experiment where he actually uses RNA and mineral glass or other mineral beads, but actually uses very specifically RNA molecules. So starting sort of, if you want to think about it a little bit up the complexity ladder. Karen, she's a mineralogist, and she's just doing some background work on what stuff sticks to minerals. Another theoretician. Kate Adamalo is an expert at making microscopic vesicles out of lipids, and so she's doing some very different experiments where instead of having 96 wells, we might have a few million little vesicles in which we have grains and chemicals. And Mike Travisano is doing an experiment where the selection is artificial selection, but instead of selecting like we do manually on the ground, picking out our wells, he hasn't started this yet, but his plan is to take grains and look at their sedimentation rate and see if the organics change the rate at which particles sediment as a way to select for some kinds of, select for higher or lower sedimentation rate. So there's lots of different approaches is the upshot. Janet is sort of our communications guru. Yes?
Do you know of any naturally occurring instances where there's self-replicating chemical films?
So the question is, do I know of any naturally occurring self-replicating films? No. So that's really the problem here, is we don't know a good way to look for them except for something like this. Yeah?
What's sacred about the surface association as opposed to free floating?
So so the question is, why do we care about the surfaces? What about a free floating chemistry? The problem with free floating chemistry is that, remember, this is about cooperation. So we have these keystone species that cooperate. In a free floating situation, they are diffusing at the same rate as the food particles. And so unless the entire ocean becomes one great big living system, it just diffuses away from one another. So you need some kind of way to keep cooperating chemical species close to each other. A cell membrane does that beautifully. A solid surface does that. There are a few other possibilities, like you could imagine maybe the surface of oil droplets, or maybe at a liquid-gas interface. But you need some kind of way to slow down the rate of diffusion of the cooperating molecules while allowing the food to keep flowing freely at a higher rate. Yes?
Did you just pick 2 days for generation
Yes.
of the air, or have you tried other?
So the question was, did we just pick why did we pick 2-day generation times? Yeah, I mean, we just picked for kind of convenience. Every day means someone's got to come in every weekend, and 2 days a weekend. So, I mean, it's like practical. It's when you have too many choices, it's hard to pick. So you might as well pick what's convenient.
Have you tried other bugs?
We not in these experiments. We haven't so far. The plan is to actually do. In the summer, we're going to do a week generation and another set of, I think, 2 days as well. Maybe 1 day. I forget whether we're doing 1 or 2 day, actually. Yes?
I know there was a lot of talk a while back about clay surfaces. Are some of the grains that you're using basically clay?
Clay, yeah. One of the 4 minerals that we're focusing on this summer, one of them is a clay. Cool. Well, a clay is a phyllosilicate, so it's, yeah.
When you use temperature to speed up your reactions, doesn't that also encourage desorption?
So so the question is, if we use higher temperatures, it will speed up reactions, but it will also increase or reduce the degree to which the chemicals are stuck to the surface. It will increase desorption. Yeah, that's the problem. It can do both.
That could change your system.
It could change the system, and we don't know how sensitive the system would be to the specific physical environment. pH, ionic strength. There are many other factors that could affect exactly what happens on the surface, and we don't yet know what matters and what doesn't.
Do you have any archaeological geologists involved in your teams that might be thinking about conditions after Earth is first formed?
So the question is, do we have any sort of paleogeologists or who can tell us about early Earth geochemistry? Our expert there is Karen Rogers. That's what she works on. And so she's the one I go to find out, what minerals to use and what kinds of solutions are reasonable. And then there's another collaborator not on here who knows a lot about making these milliuri soups and how the gases you use changes the composition. It doesn't change it as much as you might think in that case. Yes?
I was thinking about and wondering about electrical fields and magnetic fields? Because certainly at the origin of life, we know that there were quite a few ambient electrical and magnetic fields.
So the question is about magnetic and electrical fields, and I guess we ought to add, just in the general sphere, light energy. I mean, it's not quite what you're thinking, but we haven't thought about magnetic or electrical fields. We haven't played. Well, that's not entirely true. Early on, we had the idea of actually, instead of giving redox in the energy in the form of out of equilibrium mixtures, we could just stick electrodes in. But we haven't thought about magnetism. We are thinking about light as an alternative energy source to these redox pairs.
I mentioned the magnetic stuff because we found in surgery that bone growth increases in magnetic fields.
Okay, so bone growth.
And insurance companies pay for it.
Yeah, so we haven't thought about that. That's a good thing to think about. Thank you. Yeah?
Have you collaborated with any computer modeling researchers of the Miller-Urey experiment?
We have we collaborated with like modelers of like earlier
modeling of the experiment.
Yeah, no, not directly. Jim Cleves, who have really just recently started collaborating with, does do a lot of computational chemistry on these prebiotic mixtures, but not in a really geological sense. Yeah.
You made some statement about the appearance of life on Earth in relation to the age of the Earth and the formation of life. How close back to the beginning of the Earth does there able to be demonstrated that there was life? So would it be necessary to try to reproduce those types of conditions in what you're doing?
So it would be nice to. Let me show you. I actually had a slide that I then took out that actually gave these numbers. I think it's still here. I'll give you the. Oh, I totally took it out. Okay, so I can remember it, I think. So I think somebody here will probably correct me, but I believe that the Earth formed about 4.5 billion years ago, 4.54 billion years ago. And the current estimates are that probably liquid water was present at least 4.2 billion years ago. The oldest rocks that people are sedimentary, so they have the potential to contain life, that people have analyzed, are dated at 3.95 million years ago. And these ones don't have physical evidence of, you can't see fossils of microbes, but there are carbon nodules, little carbon inclusions, where the carbon atoms are biased in the way that indicates that they went through life. Because the ratios of the isotopes don't look like geological ratios, they look like life ratios. And then you have to get to about 3.8 billion years to find the first things that people have interpreted as physical fossils of bacterial-like cells. So 3.8 billion years ago, so 3,800 million years ago. If you say 4.2 billion years ago is your starting point, there's a 400 million year gap between water, roughly, and those first visible fossils. It's a long time. Yeah. Longer than the average tenure faculty member is working.
Another question?
Yeah.
Do you notice any difference in like the variations between the generations? Are they linear or are they exponential changes?
Well, see, we haven't really seen these consistent changes yet, so I can't really answer that question. I mean, I know what evolutionary theory would predict, which would predict that, once you would see nothing, and then maybe you would find that first autocatalytic set, and it might be a little step-like, but then you expect. Actually, I don't know. I'm gonna retract. I don't know what evolutionary theory predicts, but you might expect little steps as you add these new autocatalytic sets. These little cores, but we don't know yet.
Any others?
Yeah.
Are there any sort of synthetic systems that are very simple that behave in the way you want them to behave? Rather than use a soup, see if you can get something really elemental and see if it works with that. And then you could get maybe some surface chemistry and physicists and things like that to talk about how that works.
So the question, and this is actually something that we've thought about a lot, is could we, instead of, what we're doing is we throw everything in the kitchen sink in, and we hope that this selective protocol will pull out the interesting interacting systems. The alternative would be to be kind of more of an engineer and say, okay, we know that there's a simple system that will do this. Let's put that here and then see how it grows out. And we've given this a lot of thought, and we've consulted with a lot of people, but we haven't yet found any example of a system that behaves like this that is characterized at this point.
What about polymerization of some sort? You know, where you have catalysts.
I mean, there's a lot of work on polymerization of minerals. It's sort of the go-to kind of experiment in prebiotic chemistry now, is which minerals promote which polymerization reactions. And there is this reaction called the Formose reaction, which makes sugars and is autocatalytic, but it's not surface associated, and nobody has kind of figured out a way. I'm not aware of a way to make it behave the way we want it to behave, because it's a very simple, But no, I mean, we'd love to do that. And I'm going to keep asking chemists if they've got some brilliant ideas.
The key chemical might be alcohol.
We have alcohol in there, don't worry.
Thank you very much.